This is the Naked Genetics podcast, taking a look inside your genes.
This month, we're zooming in on cancer, finding out how researchers are tackling tumours in unprecedented detail. So we're at an extremely exciting time in genetics at the moment. We're able to generate a vast amount of information about the genetics for an individual, but our problem really is about sort of curating that information, understanding its meaning. Plus, our gene of the month is the guardian of your genome, rather than the galaxy. This is the Naked Genetics podcast for May 2017 with me, Dr. Kat Arney, brought to you in association with the Genetics Society, online at genetics.org.uk. You probably know someone who's been diagnosed with cancer. It steals mothers, fathers, children and friends, breaking families apart and taking our loved ones. Around the world, more than 14 million people are diagnosed every year, and that figure is expected to rise to more than 21 million by the year 2030, so it's a growing global problem. This edition of the Naked Genetics podcast is a shorter version of the main Naked Scientist podcast, Zooming in on Cancer, covering everything from radiotherapy to building a Google Earth for tumours. You can listen and download now at nakedscientist.com/podcasts. Before we find out how researchers are delving into their genetic toolbox to study and treat cancer more effectively, I want to take a step back and find out exactly what cancer is and why we get it. And who better to tell me about that than Dr. Emma Smith, Science Communications Manager at Cancer Research UK.
Normally cells are very highly controlled. They grow only when your body needs them and when there are signals to do so. Now cancer, some of these signals, some of the genes in the cells have gone haywire, and for some reason the cells start dividing and multiplying out of control, and then you have too many of a certain kind of cell, and that's when you get a tumour forming.
And are we talking about cancer as one disease? There is, the big C. Is it just one kind of thing?
Absolutely not. We are made up of hundreds of different types of cells, and cancer can appear in lots of different tissues, the bowel, the lung, the pancreas, everything, and all these different types of cancer have different characteristics. They're caused by different faulty molecules and different faulty genes. And of course, that will require different treatments. So we absolutely cannot treat cancer as one type of disease.
What do we know about what causes different types of cancer? People blame things in the environment, people blame stress. Some people say, well, it's just in your genes, isn't it?
It's a really, really complicated mixture of all of it. Some of it is down to things that we do, ways we live our lives, things in our environment. We call these preventable causes of cancer. These are things in our environment that can cause damage to our DNA. And it's this damage to our DNA that introduces a fault, and then the faulty gene can accidentally tell the cell to start multiplying, and bingo, you've got a cancer. But of course, that's not the end of the story. Some of cancer is down to, I've seen it called in headlines, bad luck. Well, let's not call it bad luck, let's just call it nature instead. All of our cells are capable of dividing, and every time they divide, unfortunately, as much as we would like to believe it, humans aren't perfect. As a cell copies itself and copies its DNA, it can just accidentally introduce a mistake. It's not a 100% perfect process. And just by doing so, again, you can end up with a fault in a key gene and possibly cancer developing. So that's the kind of nature side of it. But chemicals and things in our environment can also introduce these DNA mistakes. So it is a mixture of both. Around about 4 in 10 cancers are, let's call them preventable. So there are lots and lots of things that people can do if they want to reduce their cancer risk.
You've mentioned that faulty genes and molecules can drive cancer. So how much of cancer is actually in the genes? Can it be inherited? What's the story there?
It can be inherited. There are certain examples where people inherit a fault from either of their parents, and this particular gene is really important, and it therefore increases the risk of cancer. But this is actually really a minority of cancers. Most cancers are simply a result of nature or just simply getting older because DNA damage builds up in our cells over time, or those things that I mentioned in our environment. So there are certain genes, like you mentioned, faulty BRCA1 and BRCA2 genes, but these form a very small proportion of cancers that we see.
So far we know that cancer is not one disease, it's many different diseases. There are complex causes, there's an interplay between the things in our environment, the things we do, and the things in our body itself. But then how do we treat cancer? Presumably it's not one size fits all for treatment either.
Absolutely not, but treatments can be broadly grouped into categories. Drugs that often make the headlines, and modern drugs are very expensive, but these drugs aim to specifically home in and target cancer cells. They're called like targeted therapies. Chemotherapy, which is one of the cornerstones of cancer treatment, it's more general in that it's not specifically designed just to hit cancer cells, but it is in some cases very, very effective. And the other is radiation treatment, so radiotherapy, and of course surgery. Put them all together, and usually patients receive a mixture as well. They don't just receive one therapy. Normally it's a combination of several different types of therapy.
Emma Smith from Cancer Research UK. Although cancer may start from our own cells growing out of control and forming a tumour, that tumour isn't solely made up of cancer cells. There's all sorts of other stuff in there too, as I found out when I went to visit Fran Balkwill, Professor of Cancer Biology at the Barts Cancer Institute, Queen Mary University of London. She's working on ovarian cancer, which spreads inside the body, particularly into the peritoneum, that's the lining inside the abdomen, and also the omentum, that's the scientific word for tummy fat.
My interest is the entire cancer. A lot of people think that cancer is a ball of malignant cells that have gone out of control, and this is true, but that's only half the story. When we think of really probably all cancers, then they are actually a mix of the malignant cells, the cancerous cells, but a whole host of other cells that the cancer has recruited and in some ways corrupted to help the cancer grow and spread. And 50% or more of any lump that is a cancer can be these normal cells that are promoting the cancer growth and spread.
So what type of cancer are you looking at?
We're looking at a type of ovarian cancer called high-grade serous ovarian cancer. And what you find when you look is that you'll find a mixture of fat cells, which are in parts of the peritoneum, and we think the cancer can use that fat to help them grow. And then you find fibroblast builder cells, you find new blood vessels, you find lots and lots of immune cells that really should, and we know they could, fight the tumour, but they don't. And the malignant cells as well. There's up to 20 or more different cell types actually in a cancer alongside the cancerous cells.
But certainly from my experience of working in a lab, you just look at cancer cells growing in a plastic dish, but obviously that doesn't capture all this diversity you're talking about.
No, absolutely, and that's the problem. Now, that's why we use mouse models of cancer, because then you can, in a living organism. But sometimes we need to have all human cells. Some of the new treatments actually have, say, antibodies that act against human cells and not mouse. And so we really need to complement the mouse models. We need to have some human models. So how do we set about this?
The human lab.
Yeah, the human lab. We are not made of plastic, and cancers are more than just cancer cells growing on a plastic plate. But with all the advances in bioengineering and regenerative medicine, we think, and many people are trying to do this now, that we can begin to build complex three-dimensional models of more than one cell type, more than just a cancer cell, and answer some of these basic questions about how tumours form, as well as what new treatments we can use.
How do you go about doing that then? How do you go about reconstructing all this three-dimensional glory of a tumour in the lab?
Yeah, well, it's with great difficulty, I think I'd say. But the approach that we've taken is to first deconstruct. And so we get from surgery, we get the omentum, which is part of the peritoneum, from patients with high-grade serous ovarian cancer. And first we found out all the things, well, many things, not all of them, but many things that we thought would be useful so that we would have a kind of template for a model, but also we would be able to validate. We would be able to say how close is any model we build to the real thing. And so we did that for quite a long while, and that's proved extremely useful and also rather interesting. And we learnt things about the tumours that we didn't know before when we were just looking at the cancer cells. And now we've taken various approaches. The most promising is really to build an artificial omentum, because in normal people, the omentum is mainly fat cells. And so we've been able to grow an artificial omentum, and we have some idea how stiff it should be from the deconstruction. We have some idea about what other cells are there. And then we are growing the tumour cells and the fibroblast builder cells, and also some other cells called mesothelial cells. And we're trying to recreate the way that a model where the tumour is first invading that omentum, and also another model where the tumour is already there in situ with all the cell types. Now, we haven't got all the cell types yet. The next challenge is the immune cells. But we've got some of the major cell types, and all from the patient putting together. And now we're just testing to see how close those cell types resemble in some of their functions and things that they make and things they do that resemble the tumours that we take from the patients.
So it's a bit like taking a house and unbuilding it brick by brick, taking everything apart, the beams, the bricks, the insulation, looking at what you've got, and then trying to reconstruct that again.
Yeah, I think that's not a bad analogy. It's very complicated. We're never going to be able to, completely recreate the cancer, obviously. The important thing now is to find questions that we cannot answer in any other way, or we need to complement our studies, say in the mouse models, and then work out what combination of cells we think can answer those questions.
So now you're managing to grow these little tumours, these systems in the lab. What's the ultimate goal? What are you going to do with them?
Well, first we need to check that they do enough to resemble the human tumour. And then from our point of view, we want to find new treatments that disrupt the interaction between the malignant cells and all those cells that they corrupt and recruit. Because if we can do that, then we break off some of the supply lines of the tumour. And we also want to use them to understand how the tumour cells and the other cells interact to form tumours in the first place. So it's understanding some basic cancer biology, but also a complementary way to the other ways we have of testing new treatments.
I look at some of the stories that are coming out of the world of tissue engineering and researchers using things like 3D printers to think about making new organs and all this kind of stuff. What would be your dream for making these kind of tumours in the lab?
Oh, well, I think that you're absolutely right. And we've just got some funding to get a 3D bioprinter. It's going to take us a while. I mean, it's not going to happen overnight, but I think it's a nice idea. I was talking about this to a load of clinicians the other day, oncologists. One came up to me afterwards and said, "Do you know when you were talking about that, I had a vision of a bioprinter printing little tiny living people."
That would be the ultimate model, wouldn't it?
Yes, unfortunately, there's something about the size of cells that make us the size we are, and unfortunately you can't make tiny people, but it's just a wonderful idea.
Maybe not tiny people, but certainly a tiny little fat farm. That's Fran Borkwill at the Barts Cancer Institute. You're listening to the Naked Genetics podcast with me, Dr. Kat Arney. If you want to find out more about how scientists are zooming in on cancer, check out the longer version of this show on the main Naked Scientist podcast feed. That's at nakedscientist.com/podcasts. Still to come, our gene of the month is a watchful guardian for our DNA. But first, more than a century ago, the German biologist Theodor Boveri put forward the idea that something inside cells was responsible for causing cancer. Today, we know that the disease is fundamentally driven by faults in the genes that normally act to keep our cells healthy, only dividing when they should and dying if they're damaged. I went to the Institute of Cancer Research in Surrey to meet some of the UK's leading experts on cancer genetics to find out where we've come from in terms of understanding the faulty genes that drive the disease, whether inherited or picked up during a lifetime, and how this might shape treatment in the future. First, I spoke with cancer genetics expert Professor Nazneen Rahman to find out why studying genetics is useful in cancer.
There are different ways in which we use genetics in cancer. The first fundamental difference is whether we're actually testing the cancer itself or whether we're testing the whole person. If you test the cancer itself, there are all sorts of genetic changes that have made that cell turn into a cancer cell. And by doing that kind of genetic testing, we can work out what type of cancer it is and what kind of treatment it might have. So that's one side in which genetics is incredibly useful. We can also look at the DNA of the whole person to see whether there was some potential reason why they developed cancer in the first place, and also whether or not there might be information there relevant to other members of the family due to inherited predisposition to cancer. So that's the other way in which genetic information is incredibly helpful in cancer.
This genetic connection raises the question of whether cancer runs in families. But as I found out from Professor Richard Halston, who spent his career tracking down genetic variations that increase cancer risk, just having several cancer cases in a family doesn't automatically mean that there's an inherited gene fault at work.
As many people know, cancer is actually a relatively common disease, so effectively, if everyone lives to a ripe old age, then you're actually going to get lots of cancers in the family, because essentially in most Western countries it's that and coronary heart disease and stroke are the major causes of death. So, and the next point is that some cancers are highly environmentally or lifestyle induced. So in examples, if you had a large number of people in the elderly who'd actually all developed lung cancer and they're all smokers, you get a sort of conglomeration of cancers within the family simply because of that. And what really alerts you to the fact there might be an inherited position in families is a cancer diagnosed at a young age, and secondly, when you have more than one cancer diagnosed in a young age of the same type of cancer. So an example is early onset colorectal cancer in a family under the age of 45, so that's quite unusual. So you've got sort of essentially defined constellations of specific cancers or site-specific cancers, but generally diagnosed at early age.
When did people first start noticing this, that it could be hereditary, and then start trying to track down what was going on?
Warthin in 1913 reported a family, so-called family G, that actually had a large number of people who were affected with bowel cancer and also uterine cancer that appeared to be transmitted in the family. That was effectively assigned to the back shelf of the bookcase, and Henry Lynch re-picked that up and revisited the family in the early 70s, and he tracked the offspring of all the people in the family G. I think he actually looked at over 120 descendants of the original people in the family and showed that they also had a very high incidence of both bowel and uterine cancer. And that then led to the definition of the Lynch syndrome, which we now know from molecular testing to be due to mutations in the mismatch repair genes. So effectively, in those cases, a mutation in one of the mismatch repair genes causes an increased risk of primarily bowel and also uterine carcinoma.
The gene faults that track strongly in families that significantly increase the risk of specific types of cancer, is that where their story ends, or do we know that they are involved in some cancers more commonly in the population?
We know that mismatch repair genes defects actually underlie around about 13% of all colorectal cancers, and they're not actually inherited, they're just actually acquired mutations. They underlie a specific form of bowel cancer, which tends to be associated with a better prognosis, and in fact it responds differently to can respond differently to drug medication.
So in people in families that have this strong risk, they have inherited a loss of these genes that normally keep cancer under control. But then in cancers that spontaneously arise in people without this inherited risk, it's something gone wrong in those cells as the cancer develops.
Yes, that's correct. And in fact, it can be this. The point about the mismatch repair aptly illustrates the fact is it's the same genes, but they're somatically or acquired mutations in those, whereas in fact other people it's inherited.
Given that there is more and more genetic data coming out, we have more advanced gene sequencing and gene analysis techniques. What would you like to see in the future in terms of using this information?
Okay, well, I think it's fair to say that really we've entered the world of the so-called 1,000 genome, $1 million interpretation problem. So I think the first point is that we actually have to interpret the data we have more effectively. That's one issue. And second, I think the other thing is that effectively record linkage is going to be critical to understanding the real, from an individual point of view, what do these actually mean. I think it's fascinating that Genome England are trying to actually put record linkage together with whole genome sequencing. That theoretically might produce a vast array of data that might be useful from a point of view of actually understanding the true functional effects of many of the variation. It's going to be a very challenging matter to understand the nuances of genetic variation, in terms of its effect on the whole person.
So the challenge is not just getting the information about the genes, but it's actually tracking that to medical records and what it means.
Yeah, I think it is. I mean, that puts everything in context. And dare I say it, the word big data always creeps in, but it is ultimately a big data challenge, and that's going to be a very significant problem in the future.
It's certainly a massive challenge, but it's one that scientists are working hard at. When it comes to the genetic changes involved in cancer, there are relatively rare, strong hereditary gene faults that significantly increase the risk of one or a few types of cancer in a family. Then there's a much wider spectrum of genetic variations across the whole population that have a less severe impact on cancer risk. On top of this, our DNA is under assault every day from things in the world around us, such as tobacco smoke, and even the fundamental processes of life at work within our cells. And it's this damage, combined with our underlying genetic makeup, that can result in cancer. Thanks to advances in DNA reading technology, known as sequencing, scientists can now delve into the detailed genetic makeup of an individual patient's cancer and even use that information to shape treatment. One example already in the clinic is the breast cancer drug Herceptin, designed to target an overactive protein molecule resulting from a genetic mistake in some types of breast cancer. But it doesn't work for every patient, because it turns out that all breast cancers are not the same. To find out how the genetic revolution is changing our understanding of cancer, I spoke to Dr. Angaraj Sadanandam, who's combining lab research with computer modelling to target tumours more effectively.
Conventionally, people were thinking that a breast cancer or colon or pancreatic cancer is one disease. And that has been tried and therapies have been given so far, and it was not successful because different people respond differently to these drugs. With the genomics improving so much, we can untangle that and try to identify the sub-disease within each cancer type and try to match them to the therapies.
So basically, all breast cancers are not the same, even though they might all start in the same part of the body?
Yes. Though they start in the same part of the body, the question is that are they starting from the same type of the cell? That is what is an interesting question. If they're not starting from the same type of cell, then they will have a different prognosis and therapeutic responses.
So by finding the key genes, the key gene faults that are driving these different groups of cancer cells, how then do you target those with treatments?
These key genes later, they translate into a protein, and the protein are the functional units of the cells. These proteins have a particular structure, and that structure defines how a cell would move or how a cell would change their shape and all those things. So we can try to, by computation modeling, and make certain chemical molecules which would go and bind at that particular structure. So that way the cells cannot get the signal further to move or change shape, so that you can control the cancer cells.
So these are very, very targeted therapies, targeting the faulty products of these genes in the cancer cells, wipe them out.
Yes, yeah. So that's the basic idea, so that these cells don't behave that way.
What do you see as the really big questions that still need to be answered when it comes to using genetics to understand tumours and how to treat them? What are still the big mysteries?
So one other major complication, now we are thinking that these patients, one patient's tumor differs from another patient, but the much more complex thing is within a patient, there are a lot of subtypes and subgroups existing because, as I mentioned, if they are starting from the stem cell, within the tumor, they can become into multiple groups. And if you take one patient's tumor and cut them into, let's say, 20 or 30 pieces, there are possibilities that if they're not going to behave 20 different ways, but at least 4 ways or 5 ways or 10 ways, that is a more complicated things. And still we are in a really early stage to understand how these changes are happening. So if we go and start treating them thinking it's a one tumor, it's not going to work.
I guess it's like looking at large families and saying, well, you've all got a lot of things in common, but when we really drill down to it, each member of that family is unique and individual.
Yeah. So between two individuals within a family, there may be a significant change to make them behave differently and also do things differently. Similarly, the cancer cells can have a very tiny changes, but those tiny changes can make them interact differently. At the same time, there are not only cancer cells within a tumor, but there are also other cells, immune cells, all those interact and make them even much more complicated, and they behave differently. That's a major challenge to untangle right now.
What do you want to find out next? What are you looking at now?
So the major challenge right now is to define the key set of genes which we call biomarkers, because these biomarkers would define these subgroups. That's my first challenge, and we think in bowel cancer, for example, we have defined those genes. And second thing is which type of technology that would take that to the clinic. The question of how to handle a lot of data given a patient coming to the clinic in a single day or two is very, very challenging. So new methods and technologies are coming which don't have to deal with thousands and thousands of genes, rather tens and hundreds of genes, so that you can give the answer to the patient within a day or two.
And finally, if you imagine, say, 5 years into the future, what would you like to see cancer testing and cancer treatment look like based on the kind of work that you and your colleagues are doing?
So when a patient enters into the clinic, that the blood and the tumor material from that patient would be taken to ask the question that what kind of subgroup they belong to. And if that is the case, with existing drugs available, what would be the therapy that they can go for? So, for example, patient A entering in, okay, you have this particular genetic type, so you are qualified for drug X. And patient B, you go for drug Y. And that's the way I'm thinking 5 years from now. If not completely achieved in a subset of patients, we could probably find the way to go in that direction.
Advances in gene sequencing have the potential to target cancer in a more precise and more personalised way than ever before. But this new wealth of data brings its own problems, as Nazneen Rahman explains.
So we're at an extremely exciting time in genetics at the moment. We're able to generate a vast amount of information about the genetics for an individual. But our problem really is about sort of curating that information, understanding its meaning. And there are lots of different ways in which we're not able to use that information in the best way possible. And so one of the things that we're currently doing together with a lot of different people across the UK and indeed the world is an initiative called the Transforming Genetic Medicine Initiative, the TGMI. And really what that's trying to do is harness all of this information and use it to bring knowledge and hopefully wisdom so that we can make sure that we're using the information appropriately to bring the maximum benefits, but also to make sure that the information is not used inappropriately as well. So I think that's the key challenge for us in genetic medicine currently.
And how do you go about doing that? How are you bringing all this information together?
At many levels, it's really quite prosaic, and a lot of it is sort of logistically trying to pull together lots of different information and then sort of curating it. So there are 20,000 genes. At the moment, it's not entirely clear how many of those genes can be associated with human diseases and how many are not. And historically, there's a lot of unclear information that was unclear before because we were having more and more knowledge, but you have to go back now and then look at the previous things and think, oh, now we've got this new knowledge. How do we have to reinterpret sort of previous information? So at the very highest level, we'd really like to sort of go through all of the genes and say, okay, these genes can be associated with human disease and these ones are not, and be able to separate those out. And then for the ones that are associated with human disease, how are they? What diseases? What types of mutations? And then once we've sorted that out, how does that then translate into how those are going to be tested and what are the best ways of doing that? So at the heart, there are really quite simple questions which until now we haven't had enough data to be able to answer in a sort of comprehensive and careful way.
It feels like there's a lot of sort of philosophical changes that need to happen, just the way of, instead of saying you have breast cancer, to saying you have this particular genetic subtype of breast cancer. And then there needs to be a sort of philosophical change in the way we develop drugs, the way we test drugs, the way we license drugs. How can we make that happen?
Well, I think recognising the problem is a key step, debating it. I think with this, the scale of technological change that allowed us to go from taking 10 years to do one genome to being able to do 10 genomes in one day is such a massive change that it makes the whole system require a sort of ground up, just let's think about it again and how we're going to make it work in this modern world. So I think the key thing is really recognising the issues and debating them. There are philosophical debates to be had, not least about the sort of predictive information as well. Do we want them as individuals and society? Do we want to have this window into the future? When do we want it? How do we want it? How are we going to act upon it? These are not things that we've had to think about very much. So I think that's the first thing that has to happen. But then we have to then overlay that with really some quite pragmatic solutions. And I think we might have to almost do it in a sort of an experimental way, try things, get the information, iteratively, then improve it. I don't think we can solve it in isolation and create the perfect system ahead of time, not least because everything is still changing very fast. So I would like to see some ways in which we can develop the system and improve it as we're sort of working through it. Otherwise, we'll always be chasing our tail and we may never get there.
Nazneen Rahman, and before her Angaraj Sadanandam and Richard Halston, all from the Institute of Cancer Research. As well as studying the genes involved in driving cancer and using this information to drive the development of new cancer drugs that target faulty proteins, researchers are also taking a more direct approach, investigating how to turn down the activity of genes themselves. One of them is Professor Peter Leedman from the Harry Perkins Institute of Medical Research at the University of Western Australia in Perth. He explained his ideas to Chris Smith.
We're fascinated with the concept of using part of the genome, that is the DNA blueprint, as a new therapy in cancer. And in particular, some of the solid tumours like liver cancer, pancreatic cancer, head and neck cancer and brain cancer, for which there are very few treatments and the prognosis or the outlook might be very poor.
We know cancer is a genetic disease. It's when genes go wrong and therefore cells begin to obey the wrong sets of instructions or disobey the normal sets of instructions. So when you say you're trying to use the genome to come up with a new way to treat cancer, what do you mean?
Some of these solid tumours, they're driven by factors such as receptors that sit on the outside of the cell, and they drive the cells to proliferate or grow in an unrestrained way. We're looking at that as the set of targets. The second part of this story is saying, what can we discover inside the genome or the DNA blueprint that might give us an answer for slowing down that receptor which drives the growth? And the dogma for 50 years was that a lot of the DNA was said to contain junk, so-called junk DNA. Or what's been discovered over the last 15 years is that about 2% of the DNA is really important and made into protein, but that 98% of the non-protein making DNA is really important.
Just to clarify that a bit then, so you're saying that 2% of the DNA code actually are genes, and those genes turn into proteins in cells. They're like recipes that make proteins. The 98% is really important, but how? What is the 98% making then or doing that makes it so important?
So the 98% tends to make what we call non-coding RNA. So DNA is made into RNA, that this copy, and it's that copy that's not turned into protein in the 98% of the genome, and it's that contains this rich tapestry of little molecules called RNA. And that is an area that's of great interest globally, because we believe that if we can untap some of the secrets in that 98% of the DNA so-called junk, some of them might be useful for clinical treatments.
Right, so the dogma of what's useful about DNA is it contains recipes that make proteins, that's going out the window, and what we now realise is that it might be not be making proteins, but this DNA is sure as hell making something important. It's making these RNA molecules. And what do they stay in the cell and do something?
So what we know, some of them are really small, only 22 or 23 bases long. What we found is that one of them, called microRNA-7, is particularly powerful at slowing down and inhibiting the growth receptor that drives a number of these cancers. And it does it in a very sophisticated way. It basically takes out the key receptor itself and multiple friends of the receptor that help drive the growth and promote the tumour. And what we find when we add microRNA-7 to a number of these cancers, we can stop the growth of the cancer in the test tube. We can also stop it in mice, and that's particularly exciting.
What do they do physically inside the cell to achieve that effect? Do we understand chemically what they are doing?
So what we know is that the microRNAs work inside the cell and they target the message and basically blow it apart. If that protein being a key growth receptor that drives the cancer, that now is taken out of the cell, and so the cancer undergoes what we call cell death.
So it's a way of specifically and in a highly targeted way taking down a discrete genetic message. So it robs the cell of the ability to have that genetic message that it was making too much of. Correct. To make it at all. The only problem is that would happen in every cell in your body. If you just deluged a person in this microRNA-7, they would be robbed of the ability to make that gene product everywhere. And that would be bad, wouldn't it?
Well, that's a great question. What we know about these tumours, these nasty tumours such as liver cancer and head and neck cancer, for example, is they're characterised by having lots, and I mean sometimes millions, of these growth-promoting receptors on the surface of the cell, in contrast to normal cells, which might have 1,000 or much, much lower amounts. And the principle here is to replace back something that the cancer cell used to make. And that's a really important point. Cancer cells we're looking at used to make microRNA-7 and they've lost it. And it provides this incredible break on growth promotion. And when you lose it, you provide the opportunity for the cell to grow in an unrestrained way. Now, if we add back microRNA-7, it targets the growth receptor promoting pathway and all its friends in that, but it typically will target those cells in the body that have lots and lots of the receptor, much, much more than the normal cells. And so we believe that's why it'll be better tolerated and, of course, fight the cancer very effectively.
And you can get this message into the cells efficiently, can you?
We can. There is, when we give the microRNA intravenously in model systems, it's very well taken up into the cancer. So we believe that this will be a wonderful way of treating human cancer.
Now, one of the things that cancer is notorious for is its genetic diversity, and one of the reasons why cancers respond poorly in the end to chemotherapy is because eventually you get cancer cells that have evolved and been selected out, and they have an ability to rid the cell of whatever the agent is you're putting into it to try and kill it. So is there a possibility that all you're doing is kicking the can down the road, and we're going to select, using your agent, cancer cells that can just make even more of the gene for the receptor, and you'll be back where you started?
Great question. One of the key things in cancers is they often develop resistance to the first therapy or therapies that they might be given. One of the things that we've found with microRNA-7 is that when we give it to cells that have become resistant to a chemotherapy agent, such as cisplatin, which is often used for head and neck cancer, we find that we can resensitise the cells to the chemotherapy agent by giving microRNA 7. That's really exciting because that suggests that we can rescue patients from the terrible traumas of resistance. And in real terms, when cancer kills people, it kills people because it comes back. And it comes back in dreadful areas and spreads all over the body. You don't die from the first time you get a cancer. You'd actually die from the complications of the cancer occurring, so-called metastasis or metastatic disease. And we're very excited about this molecule, microRNA 7, because of its ability to work with other agents and make the tumours resensitised to those agents and therefore hopefully improve the outcome.
Peter Leedman from the University of Western Australia, speaking there with Chris Smith. And finally, it's time for our Gene of the Month, P53, also known as the guardian of the genome. The story starts back in the 1970s when a British scientist called David Lane was working with his colleagues at the Imperial Cancer Research Fund, now Cancer Research UK, studying a monkey virus known as SV40, which caused cancer in mouse cells growing in the lab. In particular, they were looking at two proteins made by the virus, known as the large T and small t antigens, believed to be the key to its cancer-causing abilities. By trawling infected cell extracts with these proteins, in the same way a fisherman might trawl the sea with a net, Lane hooked out a small protein weighing just 53 kilodaltons. That's the unit of measurement for molecules. Writing in their groundbreaking 1979 paper, published in the journal Nature, Lane and his boss Lionel Crawford note that it is possible that this mystery protein may normally act as a regulator of certain cellular functions related to growth control. It is of prime importance to determine the level of this protein in normal cells and see if it is induced by other carcinogenic agents. The human gene that encodes P53, called TP53, was found in 1984, and we now know it's responsible for turning other genes on in response to DNA damage. You can see your own TP53 gene at work if you've ever been foolish enough to get sunburnt. That horrible peeling stuff is dead skin cells, killed as a result of P53 switching on a cell's suicide pathway, preventing the damaged cells from turning cancerous. Hence its nickname, the guardian of the genome. That's great when it's working, but the majority of cancers seem to switch off the gene or somehow inactivate the protein, effectively throwing the guardian in jail and running riot in the body. Since its discovery, P53 is now known to be involved in a huge range of biological functions, from ageing and immunity to development in the womb. Nearly 4 decades on from David Lane's first discovery, scientists are still trying to figure out exactly how it works inside cells and how to use that knowledge to tackle cancer and other diseases. One approach is to use drugs that can reactivate P53 in cancer cells, letting it out of prison so it can kill the rogue cells. Another approach is to specifically target and kill cells that don't have active P53, which should be cancer cells rather than healthy ones. So far, this is all at an experimental stage, so watch this space. And speaking of guardians, but of the galaxy rather than the genome, as far as I can tell, there are currently no genes named Groot or Baby Groot, so they're still up for grabs in case you're a geneticist in search of ideas for your latest DNA discovery. If you want to find out more about how scientists are zooming in on cancer, including a sneaky sci-fi look at what cancer treatment might look like in the future, and a radiotherapy machine that sounds suspiciously like a steam engine, do check out the longer version of this show on the main Naked Scientist podcast feed. That's nakedscientist.com/podcasts. And of course, there are all sorts of fantastic Naked Scientist shows covering everything from space junk and the stars to the internal workings of the human body and brain. That's all for now. Next month, I'll be back with more from the Genetic Society Spring Meeting, with tales of bees and trees, and also taking an incredible 3-dimensional multimedia journey into the genome. Until then, if you've got any questions or feedback, just email me at genetics@thenakedscientist.com. You can also get in touch through the main Naked Scientists Facebook page or by tweeting at Naked Genetics. Every episode of the Naked Genetics podcast is on iTunes and online at thenakedscientist.com/genetics. The Naked Genetics podcast is brought to you in association with the Genetics Society, online at genetics.org.uk. I'll be back next month for another peek inside your genes.